Good morning. It's April 20th, and this is your daily brief in science. Here's everything you need to know. A key brain aging regulator, ATP11B, governs iron handling and mitochondrial health, and its deficiency drives iron transport to hippocampal neurons, disrupts mitochondrial respiration and dynamics, and activates neuronal ferroptosis, accelerating aging. Researchers are tracing the ATP11B-YAP axis to understand its impact on brain aging, mitochondrial function, and ferroptosis, with potential links to age-related cognitive decline. Therapeutically boosting ATP11B expression rescues memory performance and reverses neuronal ultrastructural degeneration, highlighting ATP11B as a target to interrupt iron mishandling, mitochondrial dysfunction, and ferroptosis in aging brains. Loss of ATP11B triggers a metabolic shift. Oxidative phosphorylation declines while glycolysis rises, accompanied by higher lactate and compensatory ECAR and OCR changes, signaling mitochondrial dysfunction and energy stress. Neuronal ferroptosis markers rise with ATP11B loss, including lipid peroxidation and weakened antioxidant defenses such as lower GSH with altered GSSG and NADPH over NADP plus increased MDA, ACSL4, and GPX4 pathway alterations and elevated free Fe2+. A TP11B deficiency undermines mitochondrial quality control, reducing expression of respiratory chain genes, COX5A, NDUFA4, and NDUFB6, lowering complex 1 in IV activity, dropping mitochondrial membrane potential, and promoting fragmentation and vacuolization. Single-cell and spatial data show ATP11B is enriched in the choroid plexus, hippocampus, and cortex. Loss of ATP11B weakens tight junctions, raises BBB permeability, and increases Fe2 in the hippocampus and cortex. Neuronal mitochondrial dysfunction drives ferroptosis signaling, with downstream shifts in iron-handling genes such as TFR, FTH1, and NCOA4, and lipid peroxidation pathways. Ependymal cells around the hippocampus may ferry iron to neurons in ATP11B- mice, contributing to ferroptosis susceptibility in hippocampal neurons. Behavioral and cognitive deficits accompany ATP11B deficiency, including memory and social impairments and reduced survival, aligning with increased Fe2, BBB disruption, and neuronal damage. A breakthrough in cell manufacturing has been demonstrated, printing functional structures inside living human cells in under 10 seconds. Researchers achieved the first successful 3D printing inside cells using 2-photon polymerization with a non-toxic water-dissolving resin, avoiding the cell-damaging issues of earlier methods. This method enables in situ fabrication of nano to micron-scale objects directly within the cellular environment. The approach supports printing a 10-micron elephant figurine, a barcode tag, and a functional microlaser inside a single cell, with most resin dissolving within about 2 hours. Time-lapse imaging shows the printed objects are well tolerated by cells, and the prints are inherited by daughter cells during division. The resin choice is key, reducing toxicity and enabling normal cell behavior compared with previous techniques that relied on microinjection or uptake. Barcode printing expands the combinatorial tagging space dramatically. A 4x4 grid yields more than a quintillion configurations for unique cellular identifiers. This capability enables tagging and tracking at the single-cell level beyond population averages. Potential applications include intracellular devices and sensors such as levers, springs, barriers, or responsive microdevices, as well as intracellular micro-robots. Ongoing work aims to optimize the resin to minimize cellular damage and to extend safe printing inside cells, moving toward intracellular bioengineering tools that could alter how researchers study and influence cellular behavior from within. A new 2023 review ties how fast or slow your gut moves to distinct microbiome profiles and potential health effects. Across thousands of participants and multiple studies, researchers found faster transit and slower transit each align with different microbial communities and health implications. Transit time can be estimated with swallowable sensor capsules, Bristol stool scale assessments, and markers like blue dye or corn to infer how long contents stay in the colon. The findings suggest gut rhythm should be treated as a factor in research and clinical practice to tailor diets, probiotics, medications, and other therapies to individual gut dynamics. Incorporating transit time measurements can deepen understanding of diet microbiota interactions and microbiome-related health and disease across the lifespan. Slower transit, constipation, and related gut conditions show links to metabolic, inflammatory, and even neurological disorders, including potential connections to Parkinson's disease, underscoring transit time's health relevance. Adding gut transit time data improves predictions of gut microbiota composition beyond diet alone, indicating that individual gut rhythms influence disease risk and treatment responses. Faster transit tends to associate with microbiomes rich in fast-growing, carbohydrate-lean bacteria and more variable stool consistency, while slower transit favors protein-utilizing species and may reduce diversity. The study was published in 2023 in the journal Gut, with an earlier version noted as published in December 2025. A new study decodes how the tumor microenvironment is spatially organized, showing that where macrophages sit inside a tumor can be a powerful predictor of patient outcomes. Researchers led by Prof. Lai Hong analyzed 12 cancer types to map the tumor's spatial architecture and identified 13 distinct spatial niches and 56 local cellular programs. The work reveals that niche-related interactions, including ligand-receptor signaling and cell-type-specific gene expression, help define how these niches are arranged within tumors. Macrophages emerge as key players with dual roles, promoting or inhibiting tumor activity depending on their location and neighbors. These spatial patterns connect to prognosis and to how patients respond to immunotherapy, suggesting new biomarkers and treatment angles. The study emphasizes that understanding the spatial context of cellular communication is essential for grasping tumor ecosystems. Across pan-cancer tissue, the findings establish a framework of consensus structural units that can guide future research and clinical strategies. Clinically relevant niches show significant associations with outcomes, underscoring the potential to tailor therapies based on a tumor's spatial profile. By mapping the tumor microenvironment in this way, the researchers provide a reference for interpreting how local cellular programs shape disease progression. The results point to spatial macrophage distribution as a promising biomarker for predicting clinical outcomes and therapy responses. A new chemoimmunotherapy combo shows striking early results in non-bulky, early-stage classical Hodgkin lymphoma. In a Phase 2 trial with 154 treatment-naive patients, the AN+AD approach, brentuximab vedotin with nivolumab plus doxorubicin and dacarbazine achieved high complete responses and strong short-term disease control. Safety data indicate most patients experience some treatment-emergent adverse events, with 44% having grade 3 or higher events. Peripheral sensory neuropathy was common but largely low-grade and resolving, while immune-mediated events occurred in 22%, including thyroid dysfunction and rash. There were no cases of febrile neutropenia reported. Notably, most patients did not require consolidative involved site radiation therapy, suggesting potential toxicity reduction versus standard regimens. Limitations include unrecorded steroid doses, possible unreliability of interim PET as a biomarker in nivolumab-containing chemo, focus on non-bulky early-stage disease limiting broader applicability, and relatively short follow-up for long-term outcomes and late toxicities. End-of-treatment outcomes were strong. 92% complete response, 96% objective response, and 96% maintaining complete response at 2 years, with consistent results across favorable and unfavorable subgroups. Source: Blood, led by Jeremy S. Abramson at Massachusetts General Hospital, with disclosures of sponsorship from Segan, Tacta, and Bristol Myers Squibb, and various author affiliations. Overall, AN+AD shows excellent efficacy and safety in non-bulky early-stage CHL and may allow omission of consolidative radiation, potentially reducing toxicity while preserving disease control. 2-year progression-free survival was exceptionally high, about 97% in favorable disease and 97 to 98% across PET response groups, Deauville scores 1 to 3, indicating robust short-term disease control. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.